Hello, friends. So, as a part of this snippet series on newer antifungals, so I did cover few newer antifungals, risafangin, and few others. So today, as a part of this series, we'll talk about few other newer ones which are not still available in the market. So Olorofim is one of them. So we'll just very briefly look into it. So this is still not FDA approved and it is still not available in the market. It is still undergoing certain research trials. As I said, friends, the context for these newer antifungals is because we have new fungi that are emerging. So the need for a fungal agent to cover the yeast species, aspergillus, and mucorales and molds. So we need one common agent to cover all these agents. So for that, we do not have a antifungal except amphotericin B if you want to claim there is one. Otherwise, most azoles cover only the Candida species. And for aspergillus, the drug that we depend on is voriconazole. No other azoles cover it. And echinocandins are good for certain life-threatening Candida and for auris. So, and most of the azoles which are conventionally available, Candida auris is resistant. So we need some newer antifungals. So Olorofim is one of them. So this is an interesting molecule with a unique mechanism of action. So this is the chemical structure of Olorofim. So not necessarily I would expect everyone to remember this, just for your visual sort of a grasp on this molecular structure. But mechanism of action is little interesting and little different. So basically, there is this pathway that is happening in the mitochondria. So there is this dihydroorate dehydrogenase, which catalyzes the fourth step in the synthesis of pyrimidine. So this happens in the fungi in the mitochondria. So there is a synthesis of pyrimidine, which are the building blocks of certain energy. So this pyrimidine produces this uridine monophosphate and uridine triphosphate, which create UDP sugars, which are the fuel, which are basically quintessential and act as a substrate. They act as a energy or a substrate or the needs for beta 1,3 glucan synthase and chitin synthase. So this possibly has rung a bell to all of us that beta 1,3 glucan synthase and chitin synthase are needed to create the cell wall or maintain the integrity of the fungal cell wall. So, and this particular olorofim inhibits this pathway. So, very simple. So, olorofim, so the key aspect that it inhibits is the production of pyrimidine. And this pyrimidine is meant to fuel and generate this production of beta 1,3 glucan synthase, which are needed for creation of the fungal cell wall. And it is inhibiting this particular pathways. And that is how this olorofim acts. And very pictorially, if you want to see, so olorofim as of now is available only as oral formulation. There's no IV formulation. So this is a nice fungal cell wall. And as you see, this pathway, pyrimidine pathway in the mitochondria, it is aborting the production of UDP and UTP, which acts as a substrate for beta 1,3 glucan synthase. And there is, as you see, disruption of this cell wall. So this is the way this particular olorofim acts. So very interesting. So the one that I think all of you can remember is pyrimidine synthesis, which is the fourth step of pyrimidine synthesis, inhibited, and uridine substrates are not produced. If you remember that, that's good enough. So the spectrum of activity, as you see, is little broad. Like it covers aspergillus, then it does cover Lomentospora prolificans, which is a new emerging fungi, and it covers coccidioids. So we do not have a azole which can cover Lomentospora prolificans or coccidioids, but it has no activity. So there is a limitation. It does not have activity against the yeast, which is the common ones we would wish to see it covering. And it does not have effect on mucorales. Mucorales is mucormycosis and these species, which is also important. So this is one of the limitations, but it has a good coverage on aspergillus. But when you look at the efficacy, there are no published report on the clinical efficacy. But there is some sort of an approval by qualified ID product design. There is some dimension which gives certain restricted approval for the treatment of aspergillus, and it gives approval for the treatment of this particular fungi. This is how they all look. Aspergillus looks like this. Schistosporium looks like this. Lomentospora prolificans, coccidioids, and Papillomaviriosis or Fusariosis. So these are the fungi. It has sought some restricted approval from the qualified ID product design. That's one sort of a group which gives sort of a restrictive approval for treatment of these fungi. So what are the ongoing studies? So there is this formula OLS study, Olorofim formula study. Basically, this study is looking at initial trials to see the effectiveness of Olorofim on these particular fungi, Scedosporium, Plomentospora prolifens, and coccidioids, which are resistant to currently available antifungal drugs. So these fungi which are resistant to antifungal, so they're looking at the efficacy of Olorofim in this formula OLS study. And there is this another study which is an ongoing called Oasis study, where they're comparing Olorofim versus liposomal amphotericin B to see its effectiveness against these aspergillus. So these are some of the ongoing studies that are happening. So if you ask what is the perspective on the future use in the treatment of mold infections, at best, we can look at Olorofim as an oral step down, because right now the drug of choice for aspergillus is we have voriconazole, which is available as IV formulation, but this particular drug is only available as oral formulation. So it may not be a great drug to start as a initial therapy for a sick aspergillus infection. So at best it can be used as a oral step down. Since there is no IV formulation, so it limits its first line usage. And there are certain side effects which are very minimal. So it can cause rise in the liver enzymes, and it can cause some nausea and vomiting. So this is all we have about this particular drug. So Olorofim, as you see, the mechanism of action is through inhibition of the fourth step of the pyrimidine synthesis, and aborts the production of uridine monophosphate and triphosphate and the sugars, and it inhibits the formation of beta-1,3-glucan, which is needed for the cell wall synthesis. And if you look at its limitation, it does not act against the yeast, which is very important for us, and it does not act against mucorales, but looks like more effectiveness against these aspergillus, coccidioids, and other drugs. And there are ongoing trials. So it may be a competitor to more voriconazole is how we can look at it, and some exotic new fungi that are evolving. So that's all we have information about this particular drug, which is unavailable anywhere in the world in commercial market at this point of time. So that's about it, friends. So I request you all to submit valuable work to our Journal of Acute Care. And of course, you can visit my website to read to this lecture. Thank you. Thank you, Windows.